Yes, sir. So welcome to this seminar in the AP 483 series. Today we welcome Professor Tom Baer. He's a professor of applied physics here at Stanford and is also associated with the Stem Cell Center. And he's focusing on bioimaging a lot of his research, but that's not really what he's going to talk about because he is a person with lots of talents. You know, he's a fellow of 3 international societies, including Optica, formerly OSA. So he's probably the one who designed this T-shirt that I have on. At least that's his organization, because he was the president of Optica or back then, OSA, in 2009, and he's also led a lot of committees on policy work. So right now, I think he is leading the Optica's International Public Policy Advisory Committee, and those are important policy fora. And so that's the other aspect of Tom's work, not only on the science side with imaging in particular, but also on sort of policy. And today, I think we're going to talk about something that is at the intersection of those 2 things, if I'm not completely mistaken, Tom. But of course, you're going to explain all of that to us. So we'll find out. So with that, Tom, I'm going to mute myself and let you take it away, let you share your screen.
Very good. Thank you.
So let me remind everybody to mute themselves at this point. Just click the mute button right now, all of you, except for Tom, of course. Thank you.
Well, very good. Well, it's my pleasure to be giving this seminar, part of the AP 4883 series. I think it's my 3rd or 4th time over the last 15 years that I've given seminars of this sort. I have always enjoyed it and mainly enjoyed giving them in person. And I'm sorry I won't be able to do that this time around for obvious reasons. But it is a real pleasure to be a part of this series. And you know, I'll be talking about some of the work that I've been doing. Let me just start sharing my screen here. In the areas of science public policy, but also in terms of some of the research I've been doing with people at Stanford in 2 of the most important challenges to our society and to the whole globe today, which is the COVID-19 pandemic and climate change. There are a couple of messages I wanted to give to these students here who are participating in the course, and that many of you are studying optics and photonics, and it's a technology that is really an enabling technology across a whole wide range of disciplines. And certainly in my career, I've participated in a whole different series of different applications of optics and photonics technology. And it's been like a continuous series of 5 or 6 year postdocs, very well paid postdocs, but it's been just wonderful to be able to learn different areas of science using the skills and technologies that I learned as a student. And I hope you will recognize that what you're learning here in terms of learning how to do science and practice science, it has a wide range of applications across many fields. Those fields you choose to participate in can be ones that have great societal impact. And that's been a theme in my career, both in medicine, now in climate. And it is an option that you have, and you can choose to direct your careers in these directions and make some significant contributions. And in fact, right now, what has happened over the last couple of years, just fairly recently, is the scientific societies and groups at Stanford have really enabled Stanford students and faculty to participate globally in international efforts. And I'll be talking about that as well as how these things came together and how you can participate in these particular effects. One of the things I wanted to mention is this is one of my favorite quotes of Art Schawlow, and it was from an APS student dinner I attended in 1978. And basically, he said, you don't have to be an expert in the field to make a significant contribution. And I think that's important to realize, is that you can use the skills that you'll be developing over the next few years and make important contributions in a variety of different fields. So that's another theme of this particular talk. So I'll be talking about both climate change and COVID-19, fairly disparate topics. And about half the talk will be on climate change, half will be on COVID-19 pandemic. And I'll be starting with just talking about optics and photonics technology and what roles it's playing in climate. And I'd like to start with this particular video. I hope it's running smoothly. And the video really shows the CO and CO2 concentrations as a function of time. And it was a model that was put together by NASA. And it's really a beautiful model that you can download, and I encourage you to do it. It recapitulates a lot of the atmospheric dynamics that take place throughout the globe. In terms of, if you look down at South America and the Amazon, you can see it breathing, and that's the daily respiration that's associated with the rainforest. There are the gears, the circulating patterns in the ocean, the Arctic vortex and the Antarctic vortex. And if you speed it up like this, you can see over the seasons, you can see the diurnal changes, which show the different concentrations, emission patterns of carbon dioxide and CO. It's really a fascinating model, but it is just a model. And here, this model was done in 2006. And if you see it actually, the model itself went up to about 400 ppm. We're already above that level at this point in time, meaning at several ppm per year based on the emission levels. So what is the issue here in terms of how optics and photonics can play a role? Well, a lot of these models, particularly when they are devoted to trying to describe the impact of climate change in a regional basis, for example, the Arctic, the sea ice in the Arctic area, they don't work very well. And it's primarily because they are not terribly well constrained. So part of what we're doing as a community of scientists and technology developments is developing a better measurement capability. The reason why it's difficult to do these models is that all the different spheres that contribute to our climate, the atmosphere, biosphere.
So just one thing, Tom, let me just break in here and repeat again that please mute yourself if you're not Tom. Everybody else will be muted at this point. I shouldn't hear typing or anything like that. Thank you.
Okay, very good. So let me just continue. There are different spheres, and all these spheres are coupled together in the exchange of energy, momentum, chemistry, and mass. And that's really what determines the makeup of our environment. And so these couplings are fairly complex, involve a number of different processes. As I mentioned, this how the atmosphere couples to the different spheres through this exchange of energy, momentum, etc. And so the difficulty is these vary by space and in time. And it is how do you determine the parameters that go into these models? A lot of the physical processes are well known, but they're not well measured enough that we can constrain the models in order to be able to accurately do predictions. So these are the types of processes that are involved in these complicated models that are put together. And the role of optics and photonics, in my view, is primarily to supply better measurement capabilities so we can further constrain these models and actually get useful regional predictions of what's going to happen due to climate change. Another problem is illustrated by this particular video, which I hope you can see. And these model predictions are inverse problems. And that is you're taking a set of measurements that you've made, and you're trying then to develop a model which predicts those measurements based on previous time periods. And I'll be talking a little bit more about that when we talk about a particular example in greenhouse gas emissions in cities. So it's like trying to predict the act of pouring the cream into the coffee by taking measurements of the turbulent behavior of the coffee at this later point in time. It's a more complicated problem than most in science. And the importance of measurement is you can't measure, you can't manage well what you don't measure. So it's important to be able to establish our capabilities, improve our capabilities for measurement in order to be able to better develop models which predict where we're headed in terms of climate change. It's with that motivation in mind that Optica, formerly known as the Optical Society, has developed a joint program called the Global Environmental Measurement Monitoring Network Program. And we're doing that in collaboration with the American Geophysical Unit. Okay, OSA, of course, is the leading scientific society in the science and technology of light. And AGU is the leading international society in terms of earth science and climate change. And we bring in, as part of Optica, primarily the instrumentation and measurement capability. AGU brings in the science, earth science capability, and it's a very useful partnership. Both international society, both are global international societies. In the case of Optica, about 55% of the membership is outside the US. And so really this can be a global effort. We've taken advantage of that. And part of what we've done is encourage the development of what we call GEM centers. And these centers involve people like us who are doing the measurement, instrumentation development capabilities, AGU representing climate modeling, the actual in the field measurements, industry, international standards, groups that take data and package it in a way that can be used to help formulate government policy. So these are the GEM centers, and we have one here at Stanford. It's a joint collaboration between Stanford and Cal, and I'll talk a little bit more about that. And we have half a dozen GEM centers located in universities around the world, as well as discussions going on with another dozen or so universities where they are interested in establishing comparable centers. Optical plays the role of networking these centers together and provides them with staff support for meetings and publications, etc. Optics photonics technology plays a major role in environmental sensing. Satellite imaging is a major role in terms of developing these global models. Airborne platforms using laser spectroscopy methods measure the chemical makeup of the atmosphere. LiDAR techniques are used to monitor emissions from volcanoes and from other non-anthropogenic sources that are important in the makeup of the atmospheric chemistry. Point sensors that involve optical components are used to monitor greenhouse gases and toxic gases and pollution gases within urban environments. And just as important in terms of determining the atmospheric constituents is the interaction of the atmosphere with the ocean, which is determined by a series of floats that I'll describe in more detail, and lots of optical instrumentation packaging within these Argo system floats. So optics plays a major role. What I'm going to be doing is just highlighting several projects that I've been involved with or are ongoing at Stanford as highlights of this. But of course, there's a whole range that would require more like a course rather than just a seminar to go through the whole repertoire of optical applications. One I want to highlight is one that I've been involved with very extensively over the last 2 years, which is a program that we started as a part of the GEM network, working with the GEM Center here in Northern California and one in the UK, centered in Glasgow, Scotland. And it was an integral part of the COP26 event that took place just a few months ago. And what we focused on in that particular case was a collaboration between the GEM Center in Northern California, in particular work that's being done at University of California, Berkeley, and the city of Glasgow, where the COP26 event was held. And we focused on looking at how cities are contributing to the loading of the atmosphere with greenhouse gases, and how they are going to be an absolutely key portion of the solution. So this is a brief video we put together that kind of explains in detail. Hopefully it will run what we were doing as a part of COP.
Cities are responsible for supplying a safe, healthy, and sustainable environment for their residents. Since cities generate 70% of greenhouse gas emissions, they have an additional challenge of helping reduce the impacts of climate change and help their nations meet greenhouse gas emission goals under the Paris Agreement. Two leading scientific societies, Optica and AGU, are working to help cities reduce these emissions by providing them with the data to make sound, evidence-based decisions. Highly accurate, location-specific climate data is necessary for governments, communities, organizations and researchers around the globe to make the political and business decisions that are needed to halt and reverse the impact of climate change, as well as adapt to it.
The Global Environmental Measurement and Monitoring Urban Air Pilot Project benefits global research and demonstrates how universities, leading scientific organizations and governments can work together towards a common goal.
This innovative program is deploying state-of-the-art sensors around Glasgow to monitor greenhouse gas emissions and pollutants in real time and will produce more accurate maps of the city's GHG inventory and air quality. The GEM initiative is connecting experts from around the world to form regional centres that create and share new measurement technologies and climate models. Sensors can be deployed in any city around the world and provide localised information. And the more cities that participate, the better the models. Learn more at geminitiative.org.
Well, hopefully that video clearly.
As I say, as of today, we are India's fastest growing residential.
Again, if people could mute their microphones, that would be helpful. So the GEM initiative, again, organized this summit at COP26, and it was a joint effort between a number of governments, the state of California, the city of Glasgow, Scottish government, a number of universities, and measurement labs, the Met Office in the UK, private companies, et cetera. This is all organized under the rubric of the GEM initiative sponsored by AGU and OSA. And it resulted in us having representatives from cities from over 41 countries throughout the world, so it had very much a global reach. And several hundred participants were part of the virtual summit that we had. It was actually virtual and in person. It was based on technology developed by Professor Ron Cohen at UC Berkeley. And he has established a platform for measuring the gases listed here, and has installed about 100 of these in a dense array within the Bay Area. These particular units incorporate a number of different optical devices in them. The CO2 device that is measuring the CO2 level is a MEMS-based optical interferometer that measures on and off resonance of the CO2 line and allows a very accurate ppm measurement of the CO2 levels. And also within there is a very inexpensive device for measuring particulates, which is an important part of the diesel emission that are rampant in cities. The CO2 device that I mentioned here is about half the direct product cost of this. Is about several $1,000s for that particular unit. And this is one of the needs in this area is very much cost reduced CO2 and methane systems for measuring down at the ppm level for CO2 and the ppb level for methane. This is an area where I think optics photonics technology could be a significant contribution. We really need to reduce the cost of these devices so they can be effectively deployed in these dense arrays in cities in order to be able to determine where the emissions are coming from in the cities. So as a part of the COP26 event, we deployed 1 to 2 dozen of these sensors throughout Glasgow and demonstrated how these could be integrated into a series of measurements and models that more accurately predict the emission sources that are within the city environment. And the philosophy behind this is that a system, which is called the Beacon 2 system at Cal, provides the greenhouse gas measurements. These go into an inverse temporal and spatial model. And you iterate that model to get agreement between the model and measurements so you can trust it. You can use these to do future predictions and then use those future predictions in order to determine, combine them with economic and social policy analyses in order to be able to really guide the appropriate policies to put in place for cost-effective means for reducing greenhouse gas emissions within the city environment. Again, cities contribute about 70% of the total GHG emission, so they're going to have to change their behaviors and their technologies in order for us to meet our goals. This is some data. I'm not going into a lot of detail here, but this is data that Ron Cohen took over the last couple of years here in the Bay Area, and he took it before and after the shelter-in-place and isolation policies that were part of what was involved with COVID-19. You can see a dramatic reduction in the day-to-day CO2 emission, and that's because of the reduction in traffic, of course. But you can also, if you follow this throughout the year, see the different contributions of photosynthesis associated with going from winter to spring and summer here in California, as well as changes in the average speed of the traffic, where the cars get more efficient as they travel more quickly along the highways, and a variety of other issues which can then be directly implemented into models which can help guide policymakers to make the appropriate decisions in order to meet the reduction goals, which are going to be very significant changes for the majority of cities. There's some other work that's going on here at Stanford that I wanted to mention. Leo Holberg, who's in the physics department, is doing some nice work trying to develop low-cost methods, optical methods for measuring methane using direct IR absorption and interband cascade lasers. He also has some work going on and integrated path length measurements using direct emission or absorption lines measured using the sun as the emitter. Other work going on here in methane is some very nice work done by Rob Jackson, and he is using infrared imaging and again the sun as the source here in order to be able to image leaks in real time from a variety of different sources. And this is one of the major contributors to the increase in methane over the last decade or so, is the increased mining of natural gas. Beyond just atmospheric, as I said, there is a whole series of different spheres that contribute to the makeup of the atmosphere. This particular chart shows the different contributions. And I want to point out in particular, hopefully you can see it, if you look over in the right-hand corner at fossil fuels, those generate about 6 gigatons per year of CO2. And if you look at some of the other ins and outs here, in terms of the ocean, there is almost 20 or 30 times more transfer of CO2 in and out of the ocean than there is emitted by fossil fuels. The same thing for photosynthesis. So there are a number of different coupled spheres here. It's not just the sources that are important, it's also the sinks. And the ocean is the dominant sink for CO2 throughout the world. And the issue is that the processes that are involved, which is a direct gas exchange with the fluid, the ocean seawater, as well as photosynthesis. And it's primarily the photosynthesis in the ocean, which captures the carbon, very similar to the photosynthesis on land, but there is more photosynthesis present within that captures the CO2 within the ocean than there is on land. The issue and the challenge is that this is a sensitive function of both spatial and temporal, as well as depth within the ocean. And understanding the dynamics of this is critically important, because as we dump a lot of plastic into the oceans, as we change the pH and as we change the overall temperature, and because of all of this, the salinity, the temperature rise, what impact this is going to have on the net primary production, which is the sequestration of carbon in the ocean, needs to be predictable, and we do not have the necessary measurements to be able to do that. They are currently being deployed. This is the Argo system. This is a series of submersible floats that are satellite linked, that reside on the surface, then go down 6,000 feet, and then take measurements. The first generation basically did salinity and temperature and position, and then come up to the surface and transmit that to by satellite, and are able to generate very interesting data for the first time, showing the thermodynamic sink that the ocean is involved with as a function of time and season. This shows what happens on the surface on the left and deep down about 6,000 feet, 2,000 meters on the right, showing the difference in the thermal sinks associated with the different water levels. This was the first generation of Argo floats. The next generation includes a whole raft, about a dozen different instruments. Almost all of them are optical instruments. And these involve really the critical parameters necessary to predict what's going to happen in terms of the net primary production, in terms of oxygen, the CO2 levels, which determine the pH, the chlorophyll concentration. Nitrates, phosphorus, and carbon are the primary nutrients, but also magnesium and iron are basically fertilize the net primary production. And all of this needs to be measured as a function of depth, because it varies spatially across the globe as well as by depth. And this is what these next generation instruments, many of them, the biogeochemical Argo involves optical instrumentation directly. This is, for example, a UV spectrophotometer that's incorporated into the Argo float to measure the nitrate concentration. This is where it's located in the float. Nitrate is a fairly complicated spectrum when it's in salt water. It has interfering substrates due to bromine and due to just the sodium chloride that's in the water, as well as chlorophyll. So you need to do a spectral decomposition to get the nitrate out, but you can do that to on the order of 1% or 2%, and you can do that now as a function of depth. And these biogeochemical Argos are being deployed again in a global network to measure this globally. Nitrate is not only important in seawater, it's also important in fresh water. This is from the New York Times. And this is a homestead in the Central Valley in California, where they can't drink the water because of nitrate pollution caused by the agricultural runoff within the Central Valley. So nitrate measurement also is important in freshwater. And these nitrate measurement devices based on spectrometry are, again, several $1,000s each. And so the technology is too expensive. Once again, if you go back here, what we need are broadband sources really that emit between 200 and 300 nanometers in order to get the absorption spectrum, be able to do the spectral decomposition and pull out the nitrate concentrations. This is being done. There are a number of groups that are deploying these instruments that were developed locally here by Baer, the Monterey Bay Aquarium Research Institute, by Ken Johnson, who gave a talk this last year at Stanford as part of the climate series that SPRC sponsored. So that's nitrate measurements locally in California. The other area of interest, some wonderful work being done by Rosemary Knight, who's part of the science effort here at Stanford. And this involves looking at groundwater within the California region. And this is a radar altimetry study of the levels, change in levels associated with different areas of California. And it shows those areas in Southern California where they've been depleting so much of the groundwater that that over the last 5 years, the surface of the earth is actually depressed 4 feet. So it's going at a little bit less than a foot per year, just a phenomenal change. And this is due to the extraction of groundwater. On the left, you see the Sierra snowpack, and during the springtime, it melts. And on the right is data that shows the runoff and the magnitude of the capture of that runoff by reservoirs in the different regions. See, we capture most of that runoff in the north, and in the south, they don't get as much, and that's why they're depleting the water down there. This is the work that is being done by Rosemary, that she is pioneering. This is essentially a half of the Helmholtz coil, a gigantic Helmholtz coil that gets lifted into the air. What you see being lifted here on top of the coil is an actual generator that generates a huge amount of energy, stores it, and then releases a large current burst through the coil. This generates a transient magnetic field, which then induces currents within the ground. And these these currents can generate their own magnetic field. They induce currents, and they can then be measured as a function of time delay. And from that, you can get the conductivity. And from that, you can determine the makeup of the ground, which consists of bedrock, clay, sand, and gravel. The water is stored in sand and gravel for the most part, but as you pull it out, you also extract water from the clay, and that's what causes the subsidence. And these interconnections, these basically underground lakes of gravel and sand, are connected. And one of the challenges then is to map these well enough so that we can use and store the surface water and store that in locations where it will percolate underground and then recharge the groundwater. The state of California is sponsoring a large study of the Central Valley using this technology that has been applied here by Rosemary's lab. She has some wonderful TED Talks and videos on YouTube, which I encourage you to take a look at. It's really quite inspiring research. So those are some of the activities that are going on here at Stanford and globally and in the climate change area. So this is the next half of the talk, which I'm going to switch gears at this point. I hope you don't get a whiplash from this, but I'm going to start to talk about now work that's been going on here at Stanford and around the world, once again, in the area of the pandemic response. This has really been one of my primary areas of focus over the last 2 years. I hadn't anticipated that I'd be moving in this direction, but the opportunity arose to work with a variety of people through Optica and through work that's been organized here at Stanford. And I'll be describing that impact. I started on this because Optica, who happens to be my daughter, who is such an expert, and she's at University of Massachusetts Medical School. She has done a lot of research in tuberculosis. And so she and I joined forces in order to be able to write a cogent article and webinar on this particular topic. She's worked extensively with the instrumentation used in diagnosis of COVID-19 and understanding its molecular biology. I've used that in previous research, but she's really the expert I relied on to put together this information. Another area of involvement that I've been quite extensively involved with was organized by Manu Prakash here at Stanford. He has put together some remarkable consortia of people. Two examples of the N95 DCON and the India COVID SOS.org. These are groups of hundreds of clinicians, engineers, and scientists, public health specialists that he has organized into teams that attack different problems having to do with the COVID-19. He's done a remarkable job of leading these teams, and they truly are international global teams that have had direct impact. He's influenced the CDC policy, NIOSH in setting mask standards, PPE standards, et cetera. He's done just a spectacular job of organizing this, and I've worked with him on aspects of N95 decontamination, as well as some of the efforts that he's done, specializing in developing new technology and delivering it to India during their last surges. So optics and photonics plays a major role in almost all areas of the different elements that have been put together in the COVID-19 pandemic. Again, I can't be exhaustive on this, so I'm going to pick out some highlights of where I think there's been some tremendous impact, and also areas that these two organizations I mentioned have been involved with. In the early stages of COVID-19, the diagnostic procedure is indicated here. You waited until you felt bad, basically, and had one of these symptoms on the left-hand side. You then went into had a test done, and if you were positive, you went home and isolated, or if you had severe symptoms, you went into the hospital, and they monitored your progression of the disease using a number of different instrumentation, if it was severe. So this is what was done previously. What was not realized was that you can be infectious and be asymptomatic, and it's now beginning to be recognized, particularly with the Omicron variant, that that's one of the dominant areas of transmission, is you don't know you're infected, so you don't know to get a test. So what is needed is to be able to switch from a diagnostic approach where you wait for symptoms to be present and then test, to surveillance monitoring. And this is what's indicated here, is you don't wait for the appearance of symptoms. You undergo periodic testing, and this is what people are doing now using the rapid test. And then if they are positive, you go in and get a secondary test to confirm it, and then you go through this process again of monitoring symptoms to go ahead. So there's really a switch here from diagnostic screening where you have symptoms to surveillance screening. And this demands a whole nother level of cost structure and availability and of accuracy. And we don't have these things at this point in time. And I think optics photonics has a major role to play in developing the next generation of screening technologies. So that what you're all familiar with is how the samples are acquired. I think most of you have done this. I certainly have multiple times in terms of nose swabs. And then the gold standard is still polymerase chain reaction, which is indicated here. And this is an optical instrument. It's basically an instrument that uses primers, which are designed to be complementary amino acid sequences, about 15 to 20 base pairs, that identify and bind specifically to the RNA, or really it's the RNA converted to DNA that are present in the viral samples in the nose swab. When these primers are bind, they are then amplified by enzymes. And each factor, you get a factor of 2 amplification as you go through a thermal cycle from about 65 degrees to 95 degrees. So every time you go through a thermal cycle, you get a factor of 2. And the way this works is that you monitor the fluorescence. So every amplicon either releases a fluorescent substance into solution or activates a fluorescent substrate through intercalation into the double-stranded DNA that's now part of the amplification process. And the way the PCR works is that the buildup of fluorescence is totally dependent upon the initial bolus of RNA in the sample. So what they do is they set an arbitrary threshold, which is usually a fraction of like 10 to 20% of the saturation point after you go through 30 or 40 different amplification rounds. You just run out of amplification reagents, basically. And this CT crossing threshold really determines the magnitude of your initial infection. And so this is a quantitative measure of the initial amount of virus in the sample. And this is one approach towards doing very sensitive and very specific detection of COVID-19. The specificity comes from the use of nucleic acid chemistry and the use of primers. And the sensitivity comes from the amplification and the use of fluorescence as the monitoring parameter. So there are other amplification techniques, and this is actually what Stanford uses, is what's called the LAMP technique. It's a loop-mediated isothermal amplification. It's a different way of amplifying. It uses primers just like PCR, but it uses an isothermal amplification process. And in this case, in most cases, it just measures a colorimetric change. And that colorimetric change is really just indicates either yes or no. The color change comes from the change in the pH of the reagents as they're being as you go through the amplification process. So it is a yes or no process. It's a digital. It's not an analog like PCR. This has been shown to be every bit as sensitive as the PCR process, and is a very reliable way to get high sensitivity detection. Again, it doesn't tell you how much you have in your initial sample. There's some very nice work being done again by Manu Prakash, who I mentioned earlier, in a program that he's called the SnapDX. And this is an attempt to develop actually a real-time assay similar to the PCR assay, which will give us an indication of what the bolus or what the level of infection is at the time of measurement. And this is important because you need to have both sensitivity and you want to know the extent to which the virus is present, because if it's low enough, the people may not be infectious and they can start to go about their normal activities. There are other methods for measuring the presence of disease, which are quasi-optical because they use a colorimetric change. And these are the antigen tests. These are the most common tests that are being used today. And the problem is they aren't as sensitive as I think they need to be to allow you to detect COVID-19 concentration levels at levels where you're not infectious. With CT levels, basically anything below 30, a CT crossing threshold of 30 is considered to be a true positive infectious level. These really start to detect COVID-19 in the low 30s. And so they're not terribly sensitive. They will tell you if you're really infectious, but it's not clear at this point that to catch the disease at an early enough stage that you're able to prevent spreading it to another person. The other problem with the Omicron is that it's got a doubling time of about 2 days, so that unless you measure every single day, you're not going to be able to track the disease as it progresses, even asymptomatically, and prevent that person from infecting other people. So there really is a need for new technologies, quantitative technologies that are highly sensitive, highly specific. I think what Manu is doing and what Optics Photonics can play is to develop very cost-effective platforms that allow surveillance monitoring in the house like the current antigen tests do, but more specific and more sensitive. Other areas where optics and technology is playing a role is pulse oximetry. Here are a couple of slides that indicate different aspects of pulse oximeters. These are very inexpensive devices. The device I'm showing here are about $15 each. They can be purchased over the counter. To me, this is the epitome of what optics and photonics technology should be developing and should be delivering. It basically consists of 2 LEDs, an infrared and a visible LED and a detector, and then a system on a chip, which is a display. So there's one integrated circuit and 2 small LEDs, integrated LEDs in the detector. These are fingertip devices. They've evolved over the last 30 or 40 years from benchtop to handheld to these fingertip devices, and you can get these over the counter. I show the one on the left, that's my hand, and you can see the readings don't agree. The top reading is the the O2 sat reading, which is really determines how well your lungs are working. And if you go below a 90% oxygen saturation limit, you really should be going to the emergency room and getting checked in to a hospital, because that indicates the development of some pneumonia. Not all over-the-counter devices work well. And Michael Lipnick, who we work with at the UCSF, has done a wonderful job of characterizing these over-the-counter. Some of them are remarkably accurate, others are not. I encourage you to look at his website if you're interested in getting these things in your house. I think everybody should have these at this point in time. The way these things work is they look at the oxygenated and deoxygenated hemoglobin, and they look at it at two different wavelengths where they cross over, where the absorption crossover between these two states of hemoglobin. And the hemoglobin is what delivers oxygen to the cells. And so again, it really just measures the efficiency at which your lungs are operating to get oxygen to the cells. The way these things work, and the reason why they call pulse oximeters, is they're really trying to measure the arterial blood. That's the blood that comes from the lungs and goes to your muscles, to your tissue. That has a pulsatile form associated with just your heart beating. The venous blood does not, and that's the blood that's been depleted. You want to know the O2 sat of the arterial blood. And so what they do is they synchronize the measurements. They then measure just the AC component of the signal, and from that they can get an accurate determination and distinguish between the arterial and the venous blood, and that gives you the O2 sat. So this is how these units work, and they're all basically integrated. All of that signal processing is integrated into the system on a chip that I showed earlier. These things are, of course, now available in your Apple Watch. And these work by reflection rather than by transmission, but it's basically the same principle. These are very useful devices for being able to monitor progression of disease in COVID-19. Beyond that, those particular applications of optics technology, some of the most important applications also involve determining the genetic sequences of the COVID-19 variants. And this involves what I show here is a flow cell. And this flow cell consists of about 500 million different spatial locations, each of which has a unique clonal nucleic acid sequence associated with the sample. And these clonal islands change color as you go about synthesizing the DNA that's present within those clonal islands. And each time you put on another base pair, the color of the dot changes. This takes place over a few hours. It involves this sophisticated imaging apparatus, which allows you to look at, as I said, 500 million different sequences simultaneously. So this is how the human genome has been sequenced, and now you can do this in a whole human genome in a couple hours. And it's the same optically based technology which is used now to study the variants that are associated with different COVID-19 pandemics. And this is extremely important because it allows us to sort of chart the phylogeny of the viruses as they've occurred in different spatial locations, and carefully chart how the mutations have taken place. This has been going on really since the beginning of the pandemic, and it's this sort of global network of monitoring capability that needs to be put in place in order to best respond to the next series of pandemics. So those are some of the areas, again, where optics photonics is playing a key role. One of the areas that I've been involved with is looking at how we can prevent the spread of disease. And that is, of course, through the effective use of personal protection equipment. The initial thought was that the COVID-19 was spread primarily by fomite transmission, which is touching surfaces and then touching your face. It has since been determined that almost all transmission now occurs by aerosols. And a lot of the work that was done early to demonstrate this was done using dark field measurements associated with aerosol transmission. And this is just some of the data here that was collected early on that illustrated why.
Stay healthy.
Great.
Stay healthy.
Stay healthy.
That's without a mask, and you can see the particles. And this is with a mask.
Stay healthy.
Effectively, there's no particles emitted. This again was done with the dark field.
Stay healthy.
And the efficiency of the masks have been evaluated, and this is part of the N95 decon effort in a number of laboratories around the world, where we built these homemade systems that actually use the same air quality particle detectors that I demonstrated that are in Ron Cohen's instrument that measure particulates in the same range for the COVID-19. So these are very, very sensitive devices which can be effective in judging the relative efficiency of various masks. So masks are important in preventing the transmission. How does the virus actually get into the body once you've inhaled it? And this is really beautiful work done by a group in the Nolan lab at Stanford, which used imaging devices to study the molecular biology of the virus and how it enters into the pulmonary system. It uses a technique that was developed by Gary Nolan and his group here at Stanford, which is Codex imaging platform, which allows multiple stainings, multiple antibody stainings of tissues to be able to unravel the complex molecular biology that's involved in the infection process. What he discovered was that the SARS-CoV-2 virus wants to attach to cells using the ACE2 receptors. Well, it turns out in the nose and in the bronchial system, there are what are called ciliated cells, and these have hairs on them, which are designed to trap small particles and prevent them from going deep into the lungs. These cells, it turns out, co-express ACE2 receptors. So you've got the perfect storm. You've got the cells that capture the viral particles, as well as the exact receptors that they need to be able to enter into the cell. And so this is exactly what the virus needs in order to be able to be so infectious. So what Gary Nolan showed was this co-expression, that the ciliated cells have very dense ACE2 receptors, and this is the entry point into the cell. And this is why masks are so important. ACE2 receptors are throughout the body, which is one of the reasons why there's long COVID situations and problems that arise systemically, but they are primarily concentrated within the respiratory and pulmonary system. So one of the major protection strategies, including other than vaccines, is to use a properly designed mask. And this is research that's been really optimized and been performed over the last couple of years and involved this N95 decon group that I mentioned earlier. And this group has looked at a variety of different methods for evaluating masks as well as for reuse of masks. And this turns out to be quite important in limited resource settings. And in the beginning of the pandemic, there wasn't enough masks around to protect the healthcare workers and for the general population. So one of the things that I got involved with was trying to come up with the appropriate methods for decontamination. And one of the methods that was proposed was to use UVC light, which is very effective decontaminant process and is used regularly in the medical area, but had not been applied to the use to the decontamination of N95 respirators. A number of people used a variety of devices that were not appropriately designed for this process. And so I worked with a group at Stanford, Bert Hesselink and Jeff Wild, to really go about carefully engineer the appropriate design of an optical instrument in order to be able to do the decontamination. The masks are three-dimensional objects, and they have a more complex geometry than many things you're trying to decontaminate, which are normally flat surfaces. So you need to have a uniform illumination. You need to be able to come at the mask from both sides and then also illuminate from all directions. There are a number of sources you could use. They typically are centered around 250 nanometers. They're LEDs, but what we chose to use was to design the systems that were based on low-cost germicidal lamps, because we were interested in having this being able to be duplicated in limited resource settings. You know, I'm a laser guy, so I'm used to working with Gaussian beams and things that look like pencil beams. Working with linear sources like this was something I had not tried to design before. I developed a very simple model, which involved using just standard electromagnetic models of linear emitters, which turn out to have a 1/r dependence, not a 1/r squared dependence. You can build a very simple spreadsheet model, which accurately predicts that a linear array of properly spaced linear emitters can provide a very uniform illumination if you design it properly. And this was modeled in both using the simple spreadsheet model, and then Jeff Wild and Bert Hesseling developed a very sophisticated ZMAX model, which incorporated just doing a ray trace model of a linear array that was placed within a reflecting cabinet. In my case, it was an office cabinet lined with aluminum foil, and it predicted very accurately what we observed and actually agreed quite well with the simple model. Those of you who have a toaster oven, it uses a very similar principle where the linear arrays are located about the same distance from the toast as they are spaced. And this provides the uniform illumination in the central region between these uniform emitters. So that's why your toast is evenly cooked in these toaster ovens, which is something I never really understood until I went through and actually did the models. So we actually built some of these, and I built these in my garage. One was an office cabinet, the other one was a garbage can, and this was from Home Depot. This is a design suggested by Bert Hesseling, which I think is a beautiful design, taking advantage of the symmetry of the masks and the symmetry necessary to get an even exposure. And this is just, these things are very easy to construct. I did this in my garage. It took about a day and a half to put together. And the idea was to design something that could be put together in the field in limited resource settings. And again, a year and a half ago, this was critical for those regions of the world that didn't have access to N95s and needed to reduce sufficient quantities of N95 and needed to decontaminate and reuse their masks. We tested these. I'm not going to go into detail here, but we tested these things using biological test strips that confirm that we were decontaminating appropriately. And I'm not going to go into a lot of detail here, but because I'm running out of time, but we then published a number of papers in applied optics that went through and detailed the engineering principles behind this. And then I worked with the Optica and the Optical Society, and we probed, we published to about 200 student chapters the availability of the designs for these devices and grants to build these devices. And about 50 of these units were built all across the world in limited resource settings. And we insisted that the student groups get in touch with their hospitals in order to make sure that they would deploy these things once they were constructed. And these groups, these student groups, did just that. And we've deployed, as I said, about 50, and they've been responsible for several 100,000 to a few million processes in terms of decontaminating N95 masks. So these, again, are just some screenshots of the different student groups and how they interface with their hospitals. So these were, again, this was a wide group, international group of clinicians and scientists, photonics, optics, photonics people who put together this particular effort, as well as staff people from Optica that made it happen. This is a group of OSA or Optica members who are involved with a new initiative, which is called the Global Health Initiative. And again, this is following in the example of the Global Environmental Measurement Monitoring, where we're gathering together multidisciplinary groups, international groups, in order to be able to develop fast response teams to these sorts of international crises like the COVID-19. So with this, I'm going to come to a conclusion here. I would encourage you, if you're interested in participating in these activities, to get in to work through the OSA or the Stanford Optical Society student chapter, and get in touch with me through them or directly. If you'd like to be involved with these efforts, we'd love to have more student participation in what we're doing, both in the climate area and in the Global Health Initiative. And I encourage you to do it. I think the skills you're developing are very valuable. If you're interested, and I hope you are, in helping to address some of these global problems, the infrastructure now exists for being able to do that. And I definitely encourage your involvement in these activities and welcome your contact if you are interested. So with that, I thank you for your attention. I'll stop at this point and be glad to answer any questions. And again, if you have further interest in this particular area, feel free to get in touch with me directly.
Thank you, Tom. Thank you for a great talk. It was interesting how you sort of we're using or talking about optical technologies both for investigating the health of the planet and for health of people, right? Optics is very useful across a broad spectrum of applications, obviously. I have a number of questions, but I think I should let other people have a chance first. So if you do want to ask a question, just unmute yourself and ask the question.
Hi, Professor Thomas.
Hi.
Really nice talk. I wonder that's for the optical detection. It seems like most of the technology is to build a good fluorescence microscope and to do a large, like high throughput sensing. I wonder, is there another way around, or we just like keep making a faster camera, like a faster scanning system?
Well, I think it depends on what the end goal is. Certainly for a diagnostic laboratory, the idea of using imaging systems of that sort and fluorescence is a very useful way to go. If we're talking about putting something in your kitchen that you use on a daily basis with high sensitivity and high specificity, it's probably not an imaging system, or if it is, it's not a very sophisticated imaging system. You really would like this to cost, $100 or so for the instrument, and then essentially, $1 for each time you use it. So it's those sorts of goals. So I think it's going to go beyond what we would normally use in a laboratory or a clinical laboratory environment, and involve some clever approaches towards using new technologies. In the case of the LAMP work that is being, the loop-mediated application work that is being done by Manu's group, we're looking at using LEDs similar to what you use in the pulse oximeters. These are actually very similar to what the 3-color LEDs that are used in Christmas tree lights, and then solar cells as detector elements, and just looking at colorimetric changes as a function of time. And the idea is to really design in from the beginning the idea that you're going to be developing this for a mass market. So it depends on what you're trying to do, and it's important to assess what the end goal is. And so I think there's going to be a variety of different technologies applied, depending upon, again, exactly what problem you're trying to solve.
Thank you.
Yeah, so if you have a question, just unmute yourself and pose it. Well, seems like there's a little bit of time, Tom, that I can ask my question. So you did show the Apple smartwatch doing oxymetry, right, for personal use, and that could be part of maybe an early warning system for COVID and other things. But so sort of an interesting question is, do you think that we're going to have sensors like that for environmental sort of chemicals like CO2 and others, or nitrates in our watches, so we become sort of an army of people uploading data from all over the planet all the time? Do you see any use of that, or do you think it's going to happen? Do you think that a sensor can be cheap enough and good enough to make that have any value, or is that just sort of hobbyist dream?
Well, I think there are some examples of that. The particle detector that I showed, which is basically costs $30 and can be interfaced to an Arduino, has been used in a community science effort called the Purple Air effort. If you look up purpleair.com, you'll see that there are thousands or tens of thousands now of these sensors around the world. And they cost about $200 to implement, and they monitor particulate levels. And again, it's an optically based instrument. It uses side scatter associated with a focused laser diode. So there are examples of technologies that have made that transition to where they could be personal devices. And I think you're going to see more of that. I worked with a company down in Southern California that has developed a wristwatch size sensor and detector for ozone. I think there's a lot of challenges. That was not optically based, that's a chemical sensor. But I think there are a number of possibilities. And I do think personal monitors of toxic gases and pollution gases, for example, is something which is an unmet medical need. And certainly in inner city environments around the world, there is tremendous geographic difference or demographic difference between different areas of the city located near high transit volumes, et cetera. And I think there is a need for this. I don't think the technology exists at this point in time, but I do think that there is the potential for the development of the technology. Will it be optically based? I don't know, but I think that clearly there is sufficient need for it that I would expect that there'll be some both commercial interest as well as research interest in developing that capability.
So if you look at it from a policy need point of view rather than a technology point of view, what type of sensor would you most like to see? A very cheap, small sensor. What would it sense? What is the chemical sensor now? What is the chemical that we most want to be able to sense in a very broad and distributed sense?
You know, it depends upon if you're looking at urban emissions, greenhouse gas emissions, CO2 and methane are the two that you want to sense. And they don't necessarily have to be portable, but they would need to be low cost and compact enough and reliable enough they could act independently. So that's in the atmosphere. I would say those are the two for greenhouse gases. Pollution gases, it's carbon monoxide, nitrous oxide, and sulfur dioxide are the primary pollution gases that cause asthma, that it would be nice to be able to have personal monitors of that device so you could determine your exposure levels. And we really don't know what health impacts are of intense short exposures. And so it would be nice to be able to collect that data. Some groups at Stanford that are doing that. So that's in the air. In the water, as I mentioned, being able to measure phosphorus, nitrate, iron, and magnesium. Those are the primary fertilization species that are responsible for that primary production. Being able to monitor that in the ocean water, and that's being deployed now in relatively inexpensive sensors at a density that's enough that will make a significant difference, I think, in our understanding. And in the freshwater, there still is, again, a need to monitor nitrate and uranium and other groundwater contaminants that appear to be impacting, significant populations, particularly in the lower income sectors of the United States and the world, indigenous community in particular. And so there are a number of areas where there's needs. I think a good half of them are going to require innovations in optics and photonics, and the other half will be chemical sensors of some kind or another. But I think it depends, again, the characteristics depend upon the application, but there certainly is a significant need for these types of devices to be developed. And that's what we're trying to do with this GEM initiative, is really identify those devices, get prototypes together and get them out in the field to show their efficacy.
Great. Thank you, Tom. I'm sure
Can I
there are
ask one follow-up?
Right.
So in my mind, there are probably two roles to go. One is to just develop very cheap sensor. Another way is to see something that we cannot see before. Which path do you think may be more beneficial, and maybe you can give some example?
Well, I think that the reason why we were working with the American Geophysical Union, AGU, is with the Global Health Initiative, we were working with comparable societies that are active in clinical medicine, is you've got to take input from those people who are developing the models, who are out in the field diagnosing patients, et cetera, and diagnosing the climate. So what you develop really needs to be guided by people who are directly in contact with the need. And so I think it's going to be a combination of both. I think clearly the easiest thing to do now is focus on those identified needs. And that's what I tried to outline here in the talk, is those areas that we can clearly see their instrumentation needs and measurement needs. As we go about developing more sophisticated models, I think there's going to be clearly new science that gets done that says we will need to measure these particular parameters. I didn't get into the Arctic and the idea of the tundra thawing, but if you remember that diagram of the different elements of the spheres that are contributing to CO2, there's a whole reservoir, a huge reservoir of carbon that's locked in the tundra. And understanding how that gets digested by bacteria and methane gets released, and who knows what else is getting released that might be a greenhouse gas, that's an active area of research. And part of the problem is we don't really have the measurement techniques that allows diurnal or daily measurements of the tundra thawing in these harsh environments. And so are we going to discover new things that need to get measured? I certainly think so. And in the health side, with COVID-19, we clearly determined that being able to sequence quickly and determine the makeup of the virus was an unmet need that luckily we had the technology around to do. So I think that it's a process of collaboration with the other disciplines that will help determine the appropriate direction for research in the optics and photonics community. And that's what we're trying to, again, establish with these collaborations with the other scientific societies in Africa.
Hi, Tom. Great talk. I had a question about your work in the carbon dioxide and monoxide detection in cities. I was interested in if you could speak a little bit to the temporal and spatial resolution requirements of such a system, because initially you were talking about identifying, key, polluters and things like that, and that seems like a very different spatial resolution than maybe can be achieved with current technologies or adaptive finding of those type things.
No, it's an excellent question. I didn't explain that clearly, so I appreciate you answering the question. Ron's approach is to develop a dense array within the city environment of sensors, spatially separated sensors. He suggests one for every 2 kilometers squared, basically, is what he is suggesting. The reason for that density is that's basically the spatial distribution of most of the emitters. So the way the process works is through this inverse modeling, as I said. The inverse model works by, you start with an a priori assessment of the emission characteristics that you expect within the city. That then gets fed into a atmospheric model. It's basically a weather model. It's what's called a WRF model. So it is an atmospheric model that describes the local circulation due to the measured wind patterns within the city. That then gives a first approximation to what you measure. And it won't agree with what you measure because usually you guess wrong in your a priori. You then do a linear regression to adjust the parameters associated with those emissions to best match what you measure. That's the inverse modeling. So the real answer to your question is the spatial resolution is determined by what is the complexity of the sources that you have in the urban environment. So you've got transportation, you've got industrial emissions, you've got heating and cooling associated with the buildings. That's on a spatial scale of a couple of kilometers squared. That determines the spatial resolution. The temporal resolution really, again, is a matter of what's going on. During the daytime, you get a heat island. You'd expect that the pollution would be worse or the greenhouse gas levels would be worse during the daytime. It turns out it's not the case, even though the emission is there, because you get a heat island effect, which then causes the gases to rise outside of the urban environment. So you want to be able to measure that, and then you want to be able to measure both sources and sinks, which vary with diurnally. They vary between spring and summer. You can actually see the impact of photosynthesis as things start to bloom here in California, in the Berkeley area, and in Glasgow. You can actually see the photosynthesis process sucking the CO2 out of the air during the daytime, and then re-emitting it the CO2 as they breathe during the nighttime. And so one of the interesting things about the Glasgow measurements was they found that they're almost at net zero right now if you include the geometric area large enough that you include the agricultural area around the city, that there's enough there that almost compensates for the CO2 emission by Glasgow. And so you could argue, you could make similar sorts of arguments for other cities. So the spatial and temporal resolution really is determined by, again, the density of emission sources that you're trying to orthogonally separate in your inverse process, and by what's going on in terms of the flux and exchange of CO2, both diurnally as well as day to day as well as seasonal, and that determines it. So what Ron has determined is that he takes measurements every 10 minutes from this dense array, and that gives him a very good assessment of what's going on during rush hour, non-rush hour, et cetera. So I think that's in the urban environment, that's the answer that you'd want. Other environments like agricultural environments, you can probably do it on a weekly basis, and it's probably just fine, and do it over a broader range, depending upon what you're trying to measure.
Thank you, Tom. We kept you for, well, Annie, you had a follow-up question?
Oh, no, I was just saying thank you.
Okay. Yeah, so thank you, Tom, we've kept you for well over an hour. So I think that unless somebody has a very burning question, we'll thank you very much again for a great talk.
My pleasure.
Thank you.